This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 111, recorded on September 3rd, 2015. Hey everybody, I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello everybody.
How are you doing?
I'm doing fine. I'm enjoying the fact that we have fall in San Diego, which is lovely. Summer ain't bad, but fall is better.
But the weather
it's
hasn't
not
changed.
fall yet. It's fall weather. It's
Really?
not fall yet.
Here it's still summer weather. It's still hot and humid. I love it. Also joining us today from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Are you in September mode?
I'm in grant writing mode, and so that's what I'm doing until October 22nd. So we're working on a big project where it's going to be a joint clinical trial with 5 other hospitals. So I'm the chief cook and bottle washer of the program. So it's either my office or a bed in psychiatry. So
very good. Well, you were going to go to ICAC.
I'm going to ICAC in 2 weeks. I'm looking forward to that.
That's the last ICAC, right?
It's the last ICAC, the 52nd Inter-Science Conference on Antimicrobial Agents and Chemotherapy.
Good job.
Say that really fast.
Good job. You've said it enough so that you get it now, right?
I get it now. And it's unfortunate because it's a really fascinating interdisciplinary meeting, and this year they're doing it in concert with another society, and so it's going to be a joint meeting for their last meeting, and I'm looking forward to it. I'm looking forward to.
Do you know which is the other society is?
I don't have that memorized yet, but I will look at
IDSA, right?
No, it's not IDSA. It's
ISA.
It's ICS.
ICS, yes.
Which is what? What's that?
No, it's ISID. It's the International Society for Infectious Diseases.
No, that's IDSA. That's ID Week, which is in October.
No, I'm looking at the page, Michael. It says ICAC ISID joint meeting.
Okay.
We're getting into an alphabet soup.
No, it's ICC. Sorry. ICC.
Yeah.
What's ICC?
That's what I'm looking at right now. ICC. We're going to get in trouble with the marketing people. Ray is going to have to bail us out.
No, he won't. We're going to show us in all our ignorance here. ICC, international what? It's got to be international.
It is international.
They should have it on. I'm looking at the front page here, and they don't even.
The acronym people are not.
Let's look about. Let's see.
Okay, well, why don't we skip that?
Here it is. I got it. The International Congress of Chemotherapy and Infection. ICC.
Okay.
But you know what the funny thing is? They left off the I. It should be ICCI. But then there's another one. They're having a joint symposium with ISID, which is infectious diseases and blah, blah, blah. You know, Michael, too many abbreviations.
I blame it all on the immunologist. I blame everything on immunologists, but that is really the immunologists love abbreviating everything.
Yeah, we do the same.
We do
the same thing. Okay, Michelle Swanson is off at a football game, so it's the three.
We miss her. We miss her.
Yeah, we do. We miss her pungent wit. So we will do this on our own.
In depth of knowledge.
Knowledge. That's right. She knows everything. All right, we have a snippet in a paper, and I'm going to give you this snippet, which was suggested by listener Junio, who wrote 1,000-year-old antimicrobial remedy with anti-staphylococcal activity. That's the name of the paper. And he says, looks like a really cool paper. Ancient Way is shown to be effective against MRSA. So this is a paper published in MBio, and that you heard the title. The authors are Harrison, Roberts, Gabrilska, Rombaugh, Lee, and Diggle. They are from a variety of places in the UK and Texas, University of Nottingham, Texas Tech, Texas Tech, and the University of Nottingham. And what they have done here is to turn back the clock, to take a time machine back to medieval days. Now, before medicine became modern medicine, there were a lot of wacky things going on. People would ground up newts and toads and treat people with them. But, sometimes it worked. And we can go back in the literature and look at some of the things they made up, some of the potions. And sometimes there's a written history of what they did, and actually see that what they're trying to treat look like microbial infections. So what they did in this paper is they went back to an ancient manuscript, a 10th century Anglo-Saxon leech book, which contains lots of remedies, and they tried to reproduce one of them and see if it works. So it's called Bald's Leech Book, an English medical text from the Anglo-Saxon period, and it has remedies for various ailments. Not all of them are microbial, but some of them are clearly microbial. And one of them in this Bald's Leech Book was a salve to treat a wen, W-E-N, or a lump in the eye, which today we would call a stye, an infection of an eyelash follicle. Everyone's had this, typically caused by Staphylococcus aureus. So they figure, well, this looks like a stye, and they're treating it with this remedy. So let's try to reproduce it and see if it will actually inhibit modern-day Staph aureus.
You're going to give him the recipe?
Absolutely, because that's the best part.
It's the best part.
But, they call this, and they say, why are we doing this? Well, one reason is we could find new antibiotics. They call them Ancientbiotics. Lovely name, right? They can give us new sources and new leads to work on. The other reason, though, is, we kind of look back on medieval medicine and say, they're a bunch of quacks, but maybe they weren't all quacks. Maybe there were some smart people who did trial and error things and came up with remedies. So,
besides, they were forced to do something.
Yeah.
Today we say we have antibiotics, we do this, we do that. We don't have to rely on our wits, but imagine being a doctor in those days and having zero in the way of, or very little in the way of an armamentarium or pharmacopoeia. You have to try it yourself. You have to be an experimenter.
That's right. And as you'll see, there's evidence for what they found that these guys did experiment. So anyway, they actually, figure one is a picture of a page from this Bald's leech book, which is the recipe. And of course, it has to be translated into English. What is it that they used in the Anglo-Saxon 10th century? What was the language? Does anybody know?
I don't know what.
I'm showing my ignorance, but
Is it Middle English?
Well, it's words that.
I think it's before Middle English. I don't think it says really.
It doesn't say. They give you the translation and the original script.
Yeah,
so
I don't know what it is, but someone will know.
That's a letter we need.
Anyway, the translation is: make an eye salve against the wen. Take equal amounts of crop leek, which is an allium species of plant, and garlic. Pound well together. Take equal amounts of wine and ox gall. Mix with the alliums. Put it in a brass vessel. Let it stand for 9 nights. Wring it through a cloth and clarify it. Put it in a horn, and at night apply to the eye with a feather. So that was their starting point, and they given the materials and methods exactly how they did this. They made 2 different recipes. They took garlic, they took onion. They used either garlic or onion, finely chopped leek leaves, and they got 25 milliliters of wine from an organic vineyard. And then they added bovine bile salts, which you can buy from Sigma, to this.
Or you can go get a gallbladder from a cow.
You could do that as well. A lot easier to buy it from Sigma. Then they mix all this together. They put it in a sterile bottle. And then they added sheets of squares of brass, because the original recipe is done in a brass flask, so they figure maybe the metal is important. The vessel, maybe the copper has something to do with keeping it sterile, right, Michael? So
absolutely,
it's quite interesting. So they have two preparations, one with onion and one with leek. They put it in the refrigerator for 9 days because these guys let theirs sit for 9 nights, and they figured it was cold because there was no heat back then. And then they test the antimicrobial effects of this potion, all right? So you add it to Staph aureus cultures and you look at the effect. And absolutely, these mixtures inhibit what we call planktonic cultures of Staph aureus, which are growing in liquid cultures, and really inhibit the growth quite well.
I was going to say, while they talk about brass, remember that brass is both copper and zinc.
Right.
It's mostly copper, but zinc also has an antimicrobial property, as well as zinc has an antiviral component as well. So they only in their discussion talk a little bit about the copper, but let's not discount the other metal that's in with the brass, principally the zinc.
So both the onion and the garlic potions work well. They also show that these inhibit the bacteria when you grow them in a model which produces a biofilm, which would be more like how the bacteria would grow in a tissue. Also inhibits them quite well as well. And then they make batches where they leave individual ingredients out because they're wondering, did these people back then actually try different mixtures and see which one worked the best? So when they leave out the onion or the leek, they still get activity, not as much. So that is important for it. When they leave out the brass, they get good bactericidal activity, but when they drop either the garlic or the wine, completely removes the bactericidal activity. All right? So garlic, wine, and the plant are crucial. Brass seems to be dispensable, but they speculate that maybe it sterilized the vessel, which maybe preserved it so that it would last longer or something like that. They also find that the 9-day waiting period was important as well. When they do shorter incubations, it's not as potent. So,
by the way, the effect is really quite strong.
It's very strong.
They really go down, 6-7 orders of magnitude. So.
Yeah.
And you wonder what the wine is doing. Could it be the alcohol is facilitating the extraction of an organic
maybe.
out of the aqueous, out of the plant phase, and driving it into solution? And one of the substitutions I would have done is substitute out the wine for absolute ethanol to see if you could get away with that and really try to begin to dissect the magic ingredient in this salve.
And the last experiment they do is they ask, do these concoctions work in an animal model? So they have a mouse model for Staph aureus infection, and they infect these mice with a methicillin-resistant strain, the so-called MRSA strains. They wound the animals and they infect them with MRSA, and then they put these salves in the wound, and in fact, they have a good impact on the growth of the bacteria in the wound. Either one, he has the one made with garlic or onion. So it works in an animal model as well as in cultures. So that's basically the paper. Very cool. They show you, and I think the coolest part, not just that it works against Staph aureus, but an antibiotic-resistant Staph aureus, but also it suggests that these medieval doctors actually tried different mixtures and came at one that appeared to work, right, by combining different things. So
one of the most striking results that they showed is that their magic mix was actually better than an ethical pharmaceutical, namely vancomycin.
That's right. Yeah, vancomycin wasn't very good.
No.
At all. It goes from about a twofold decrease in the colony-forming units.
And from 2 million down to 1 million, and their magic mix is down in the weeds, and it looks like to be about 10 to 20.
Yeah.
One of the interesting things about this is that it may open the eyes of people to the idea that chemicals in combination do different things than in pure form. In other words, instead of injecting pure vancomycin, which is what's done, or giving it to people, one is to think about other things that are required. And I think this synergism, which is kind of known for antibiotics should extend itself to a lot of other chemicals.
Yeah.
And I'm sure that what we used to call natural chemistry scientists are going to go into this and figure out what all there is in there, and it's going to be a huge number of compounds. And in combination, some of them are going to be the ones that do the job.
Well, for many years, a lot of companies, as you know, had natural products divisions that they would go outside and collect plants and microbes and soils and try and identify inhibitory molecules, but they would always purify them down to a single molecule. And so maybe a key here is, to start with a mixture like this and then identify multiple components that are synergistic. I like what they say at the end. They say not only do these results suggest that these ancient biotics were systematically constructed based on empirical knowledge, but if this is true, this predates the generally accepted date for the rational scientific method, which is about the mid-17th century, where the Royal Society was formed, and the use of sterilization by Lister in the late 19th century. This is hundreds of years before that. People are doing systematic experiments. So I think this is a really interesting paper, and it's in MBio, so anybody can access it. So thank you,
That's right,
Junio, for pointing that out. It's a very cool paper, as he says.
Yeah.
One of the interesting things is we have to appreciate that Lister and the introduction of carbolic acid to make surgery safe was only introduced into medicine in 1867, which is well after the American Civil War. And you begin to look at these things and you say, my goodness, we haven't had this knowledge for all that long a period of time.
Nope. Of course, initially it took a long time to spread because we didn't have very good communications, right?
Yeah.
But this is pretty recent. We've gotten smart only recently, I guess.
Not according to this paper.
That's true. Medieval smart too. Well, you know what? I was thinking about this. Throughout humanity and their evolution, I'm sure there were always smart individuals who were clever or intelligent or whatever. It's always the case, right? It's just a matter if their talents will come through, right? So,
sure.
This episode of TWiM is sponsored by the Education Department of the American Society for Microbiology, and they'd like you to know about the ASM Scientific Writing and Publishing Webinar Series, which is a 3-month, 6-part program that takes place from January to March of 2016. This is a webinar series intended for graduate students, postdocs, and early career scientists. And its goal is to teach you all about the scientific writing and publishing process. You'll hear from ASM journal editors about how to construct a paper, the title, the abstract, figures and legends, the review process, and much more. So, even if you are someone who's published before, things change, and it's always good to figure out and learn about what's going on. So I highly recommend this. You'll get tips for how to make a good title, make a strong abstract, the best way to select the journal, not just ASM journals, but what is the best journal for your paper to go in. You can find more information about this at bit.ly/SWPIONLINE15. SWPI Online 15, or we'll put the link in the show notes. Application deadline December 1st. You have a lot of time, but you should go to the website and check it out. It's a cool webinar series that will help you publish better papers, maybe. So that's always worthwhile. Check it out. Okay, Elio, I turn it to you for your paper.
Well, we're still on antibacterials, which is, we don't often have the two topics coincide like we do this time, but this coincides. So this is also a story with a history. It's about the effects of gallium, the metal gallium, on as an antibacterial. So gallium has been around in the literature for quite some time. I didn't trace back how long, but it's been maybe 100 years of uses of gallium as a antimicrobial and also as an anti-cancer agent. It has a checkered history, and I don't think it has yet surfaced as the cure-all, but it may belong, it certainly does belong already in some interventions, and it probably will do more. So here's the story. Gallium looks for the world like iron. Okay, that's awfully close. They're very similar in a number of ways, including their atomic radius. And you would know, Michael. You can pipe in and tell us what.
Well, gallium is one of these wonder metals that it's like its good friend mercury. It's liquid at room temperature. And the other remarkable thing is it has the same bond length when it binds in these coordination style of chemistry. So those of you who are biochemists out there will appreciate that coordination chemistry is how many of the redox-dependent enzymes in our body and biochemistry function. It uses that metal in the active center, and it has to fit in with the proper bond length and in that coordinated chemistry in order to accept and transfer electrons in what redox enzymes do. And so gallium has the same length as iron does. So you can well imagine how if you sneak a gallium in there in place of the iron, it can really screw up a redox enzyme because it's unable to pass the electrons as easily as iron because it doesn't like to have that redox moment where it becomes oxidized and reduced to the same level and extent that the iron does.
So the paper we're dealing with let me give you the title. It's called Gallium-Mediated Siderophore Quenching is an evolutionarily robust antibacterial treatment. Somewhat unusual title, puzzling title. And the authors are Ross Gillespie, Weigert, Brown, and Kümmel, all of whom are in Switzerland except one of the authors, namely Dr. Brown, who's from the University of Edinburgh. What this paper deals with is the fact that we have to go back to what iron does in the world of bacteria. Iron is an essential element, and the reason it's essential is because it's a part of a large number of enzymes, especially cytochromes and the likes, and so it's essential. Bacteria do not grow without it. Iron is largely insoluble in the world as iron 3, and therefore difficult to obtain. And the amount of iron that's available as iron 2, ferrous iron, is scarce. And so bacteria have, for a long time, as many of the listeners know, have an intense preoccupation for getting iron. And they do this generally by a very clever device, which is to send out from the surface, secreting into the medium, siderophores, that is small molecular weight compounds, which can bind iron, essentially they chelate iron. And the chelate is recognized by the cells and taken back in. So they secrete the free siderophore and bring in the iron siderophore complex. This is a big topic in pathogenic microbiology and in, I would say, microbiology in general, and a lot of attention has been paid to it. Now, the reason why gallium comes in is because the siderophores that they deal with, and they deal with Pseudomonas aeruginosa, and a siderophore that is quite common, which is
Pyvuridine. Pyvuridine.
Pyoverdin is the strongest of a variety of siderophores. By the way, siderophores come in a huge number of colors and shapes. So that are all over the bacterial world, essentially different bacteria. Well, they group themselves. There are not as many kinds of siderophores as there are bacterial species, but there's a large number. Anyhow, and many bacteria make more than one, including Pseudomonas, but this one is the main one. And they argue the following way, which is an interesting way to argue. If you are dealing with an antimicrobial agent, an antibacterial agent, which works outside the cell, you have a lesser chance of developing resistance. Think about that. Almost everything you can do to a bacterium will lead to resistance. And there are many ways in which this happens, including a nasty fact, namely that it was thought that instead of killing, if you try to kill bacteria, some will mutate and become resistant. So bactericidal drugs, which work by and large inside this bacteria, are not a good idea from the point of view of resistance. They may work fine for the sake of killing the bacteria you're after, but sooner or later, the antibiotic is going to be useless or reduced in efficiency. However, if you were to deal with the outside, you have a different problem. Think about if you put gallium in, and gallium binds to the siderophores and is not taken back in, or if it's taken back in, it doesn't do what it needs to do. Namely, it does not supply the needed iron. Okay? So this is something that you have a hard time developing resistance to. Okay, so first let's get the facts. There are a number of papers through the years which deal with the fact that gallium is an antibacterial. In fact, it is, to go fast forward, it is used somewhat or has been tried in a local sense, in a local application, for instance, in the mouth. You've run into this, Michael, I suppose, have you?
Yes, in the dental world.
In the dental world, there are there actual products used out there?
Not anymore, because we have good antibiotics. But in the ancient times, we're going to have another paper on gallium in the dental world. But today they use principally antibiotics because the toxic.
How far back does it go? Actually, I didn't look. Do you know?
It's probably in the products. It's one of these old empiric things. Gallium itself was only described in 1875, but how far it goes back in the empiric knowledge, I honestly don't know, since I don't talk about gallium.
It's been a while. Yeah, it's been a while. Anyhow, so the idea is that if you feed bacteria gallium, it's going to be an antibacterial, and that it's not going to lead to much in the way of resistance. So the paper talks about this. They have data showing that gallium, in fact, is an antibacterial agent against Pseudomonas aeruginosa. They make a big point out of certain things here, which are quite interesting, namely that the level at which it works is actually quite interesting. Too little and it doesn't work, and too much and it's toxic, which immediately brings in the point that if you were to use gallium therapeutically, you have to be extremely careful, because if you use too much, it's going to be toxic. So this is why I think it's going to be a long time before the gallium is really used in a clinical setting. It would have to be extraordinarily careful about the amount. Okay, obviously it can the uptake of iron by our cells is largely through transferrin and other proteins which bind iron. They bind gallium too. So, you give too much and you're going to poison the patient. So you have to be very careful with this. So anyhow, the paper is convincing with regard to the fact that at certain concentrations, it does work, and it works through the siderophore. It is not toxic by itself at certain concentrations. So if you make a mutant which doesn't have the pyoverdin siderophore, the gallium will still work. I'm sorry, will not work.
Will not work.
It is not toxic at those concentrations to the bacteria. It works through the siderophore, in which case it does interrupt the growth of the bacteria. Nice study. So they did that in a convincing way. I don't have any reason for disbelieving him. And then they tried it in an in vivo system. And they use wax moth larvae. Wax moth larvae have been studied as
an inexpensive pathogenesis model.
I would have thought that since Pseudomonas has been used a lot with Sinorhabdis, with C. elegans. I wonder why they didn't use C. elegans, which is not very expensive either. But here they are. Anyhow, guess what? It works. It keeps the wax moth larvae from dying, and at the right concentration, it is a good thing to use. Okay, so this is pretty much the paper. I'm abbreviating a lot, but it's about what it says. But they go through a considerable length to argue why this is not going to lead to resistance. And there's a table in the paper, but I'd like to play a game here. And I'd like to ask of you, since you may not remember exactly what's in the paper or in the table, I'd like you to come up with ways in which bacteria could become resistant to gallium. How about it? You want to raise to the challenge?
Sure.
I don't understand why that. I mean, so they talk a lot about this extracellular protein, and that's part of why it'd be hard to get resistance. But I really don't understand why that would be, because if the protein changes to avoid gallium binding, then you would have resistance. I mean, if it's so close to iron that could not happen, then that's a different reason.
Yeah, but the argument is, if you do that, you interrupt the supply of iron to the bacteria.
If in fact gallium resistance also causes resistance to iron, but we don't know that, right?
No, you don't. But you're not. How are you going to do it is the question.
That's because Elio has left out their critical experiments from their papers that they use to tell the story. And I think these authors go to great lengths to talk about they introduce the concept of public goods and private goods.
Right.
And so the siderophore is introduced to this concept of being good for everyone in the environment, because the siderophore, as long as you have the receptor on your surface to bring in iron, bacteria are very promiscuous in accepting anybody's siderophore. They want anyone's siderophore because, as Elio,
almost, not quite, but almost.
Well, they pointed out that iron, as Elio said, iron is so limited in the environment that they desperately need iron. And so if you can quench, and that's what the gallium is doing, quench the ability to quench the public good, that is, make the siderophore useless to the microbe without the microbe knowing it, because the siderophore is still taken up with the gallium by the bacterium, but as Elio pointed out, it's not toxic to the cell until you get to really high concentrations. But when you have it with the siderophore and it's brought into the cell, it's still not releasing iron to the cell, so the cell is still starved for iron. So the regulatory mechanism is still making more of the siderophore to throw out for the public good to scavenge more iron, because the cell has no way of knowing.
Well, yes, but if it makes an altered siderophore, which happens every time, right? That will now pick up iron. And let's just argue that you could make an altered siderophore that would bind iron but not gallium.
Correct.
All right. There should all right. That's the key question.
That's the key question. What's the select? And that's the key argument.
Then in that case, those bacteria with that altered gene would outgrow, right?
Correct.
Very good. You get an A.
Thank you. Thank you very much.
Selection pressure is always a winner.
But their argument is that it doesn't get much better than pyovertin.
Yeah.
And that in addition, they don't believe it. They think that gallium and iron are so close that it's very difficult to imagine a siderophore that distinguishes between them.
Yeah, but
that's what they say. That doesn't make it true.
But you can't use gallium. You'd have to use something else, and that something else would be different because.
But there's nothing as close to iron as gallium.
Yeah, but you can't use gallium. You said that we're not going to use gallium, right? So this is kind of a proof of principle, but that you'd have to use something else and that something else would be different. And I would
No,
argue
they like gallium. They think gallium.
I thought you
is the
said
right
it
thing.
would be toxic or would not
No,
be.
no, that depends.
No, at
No,
the
I
low
said that.
concentrate. At the
I
low
said that.
concentrations.
So do you think that gallium would be approved as a antimicrobial?
Well, I think that if I were the FDA, I'd give it a long look.
Yeah.
You know, this is tough because it's difficult to imagine how in a clinical setting you can really control the actual concentration in the body in a meaningful way. You know, it depends on what if somebody takes 5 pills instead of 1 pill, you know, or something like that.
Well, the true question to ask is, will gallium bind transferrin?
Yeah, right.
Because that's
but
what
it does. No, it does. That's known. It does.
Okay, so gallium will interfere with transferrin, so then it will screw
does.
up.
That's why at some concentration it's toxic.
Yeah.
It was toxic for the larvae that they used at high concentrations.
Yes, in the 200 micromolar.
So you know you have a drug which has a therapeutic threshold which may be too narrow for comfort. That's what it amounts to.
Yeah.
You know, I mean, every drug at some concentration can be toxic. Drugs are poisons, aren't they?
They are.
So, but it depends on the therapeutic threshold being very different than the toxic threshold. And if it's close enough, it won't work. But the idea, I think, is okay. The idea that this is not likely to lead to drug resistance as readily as antibiotics do, that's a valid point. And
well, here's a heretical concept, is if you use gallium in concert with an ethical therapeutic that we presently have, an antibiotic, where you impose the fitness cost, because what they showed is that gallium actually imposes a fitness cost on the microbe by forcing it to make more siderophore, because it doesn't perceive the iron concentration. It imposes a fitness cost. And what we learned a couple of TWiMs ago, that this fitness cost imposed by antimicrobial therapy can actually have a profound effect on the organism's ability, because they're asking the question, or they go to great lengths in asking about the evolution of drug resistance. If you find the right concentration of gallium that can impose a fitness cost on the population, while at the same time supplying an antibiotic that's bactericidal, you may be able to eliminate or actually change, because antibiotic resistance is looking at one drug and we're seeing the MICs get higher and higher and higher. But if we add gallium, can we get new life out of old antibiotics?
Mm-hmm. That makes sense. That makes perfect sense.
Yeah, very good.
But, you know, the thing I just don't get, this whole idea about public and private.
There was an interesting economic concept.
But it's not that. In the end, it's because gallium and iron are so similar. That's why you don't get resistance. It's nothing to do with public stuff, right?
Well,
at least
Well,
I just don't understand why a public molecule that's shared would not be subject to selection, because that bacteria is using it as well. So, but I do think the similarity.
Because you commit suicide when you do that.
Well, they also sort of tap dance around there.
Yeah, it's the iron-gallium similarity that's the key. It's not the public nature of the siderophores. That's what I'm saying.
No, it is. I think it's both things. I mean, if you interfere, any way you interfere with iron uptake, you're in trouble, right? The bacteria don't like that. They need iron. So, you know, if the gallium simply binds the siderophore and doesn't let iron in, that's antibacterial. And that is something that happens on the outside. And it's difficult to imagine how you can develop a mutant that interferes with that.
All right, let me give you a scenario. This is the last thing I'll do because I don't want to prolong it. Let's say you had an inhibitor of a siderophore that wasn't gallium, but bound somewhere else on the molecule. And it inhibited iron release in the cell, right? So this is still public goods, this siderophore.
Right.
Yet resistance can readily emerge because you can get a change in amino acids that doesn't allow this other drug to bind. So that can easily happen. It has nothing to do with the gallium-iron similarity, and it's a public good. So I don't see why being a public good per se argues against evolution of resistance. Now, maybe I'm stupid, but I don't
No,
get it.
they tap dance around that too in their opening statement about quorum quenching strategies for quorum sensing, because quorum sensing is a very similar concept where you have a public good, the quorum molecule, that is going to turn on virulence traits once it achieves its mass. And we know that the quorum sensing quenching strategies have not worked to the same level and extent because they break through and resistance does indeed happen. So I think Vincent's on the right track. It's a very interesting and provocative paper.
Yeah.
Okay.
You're right. I would suggest that listeners who really want to understand this further, in fairness, read the paper in detail. I'm not sure we've gone into enough detail to explain that argument. So there is a lot more to the paper, and reading it is probably a good thing.
It's an interesting journal I haven't read before. Evolution, Medicine, and Public Health. You guys read this journal?
I have never read a paper from this journal, but now it's on my list of things to look at because
for sure.
I was fascinated by public goods and private goods.
Yep. It's an interesting idea. All right. Thank you, Elio.
Yeah, and thank you, Michael. I'm glad you piped in.
Well, we waited an hour so Michael could teach his class. You taught a class, right?
Today was class. What
are you teaching? What class?
It's an introductory graduate student class, and we were teaching them how to work with bacteria today. A lot of these kids come to graduate school principally to focus on our immunology tract, and so they have never worked with bacteria before. So it was a really pretty basic lecture in basic bacteriology.
Okay.
But we gave them the paper to read from Ford Doolittle that's in this month's Microbe magazine. And
this is an exciting paper.
Yeah, and so we asked them if they were tree huggers or not tree huggers. And so if you haven't seen that, that's available online. The Microbe magazine is a good thing, and it's a very approachable paper on evolution and the consequences of horizontal gene transfer and what that does to evolutionary trees.
Nice. All right, let's do a couple of emails. We have one that went to Elio. It's from Noah. He writes, Dear Dr. Schaechter, I want to thank you for the effort you and your colleagues have invested in Small Things Considered and TWiM. Back when I had a longer commute to work, I listened more religiously than now, which is pretty much the only thing I regret about switching my job and reducing my commute. As a physicist turned synthetic biologist, I really need an education in microbiology that most of my peers got in graduate school, and TWiM is a great resource for people like me. Now, a question. Thermophilic organisms have DNA sequences that are higher in GC content in order to remain stable at higher temperatures. The enzymes and protein structures found in thermophiles are also significantly more thermostable. If you were to examine a conserved enzyme from psychrophile through the temperate zone and up to thermophile, what do you see in terms of its amino acid content? Are there certain amino acids that are correlated with thermodynamic stability of the protein? Do these amino acids correlate with higher GC codons? If so, do you believe that this was part of the driving force for the evolution of the genetic code? Namely, that a more thermostable codon should correspond to an amino acid that is more likely to contribute to a more thermostable folded protein. This thought never occurred to me before, but I read the abstract of this paper and it seemed to imply this concept, which struck me as sensible, but I hadn't heard it before. And he provides a link to a paper, Comparative Complete Genome Sequence Analysis of Amino Acid Replacements Responsible for the Thermostability of Corynebacterium efficiens. Thanks in advance for any thoughts. So Elio sent this off to his colleague Christoph Weigel, his co-blogger at Small Things Considered, and he wrote, in principle, your ideas are fine. Yes, GC-rich DNA has higher melting temperatures. GC-rich genomes, like streptomycetes, tend to have a bias towards G and C at the third position, and also more G and C in conserved transcription factor binding sites. However, there's a big caveat. One of the most thermophilic bugs we know today, the Archaeon Pyrococcus furiosus, grows at 100 degrees centigrade, has a genomic GC content of 40% which is 10 points less than E. coli. If I remember correctly, there is evidence that this bug employs histone-like proteins to keep its DNA double-stranded, and given its low genomic GC content, it can't have this codon bias. I could go into more detail if you want, but I'm on vacation right now in the Azores Island. I don't blame you. So this is interesting. There are always exceptions, right? There are plenty of viruses that have a high GC content, but they don't grow at high temperatures. And so it's not just GC content. I did a paper on TWiM a couple of weeks ago where we looked at a phage a virus that infects archaea that grow in extreme temperatures. And there was a good suggestion that this the DNA of this phage actually adopts a B form, which is a form, or is it A form? Sorry, it's A form DNA at high temperatures, and that may be part of why it's thermostable. And then we had a response from Noah, who he's trying to clarify his question: do all microbes that live at higher temperatures also have higher GC content? And the answer is no.
No,
not all of them. It's not just that, right? The next one's from Veronica, who writes, I was quite excited by your podcast on antimicrobial peptides. That's the one where we discussed a paper where they selectively eliminated a bacterium from a population, right, Michael?
And it was the dental paper that we did a couple of podcasts ago. Since
I studied bacteriocin usage in bacterial vaginosis prophylaxis as part of Masters. So in response, bacteriocins have been defined that inhibit BV-associated pathogens while having little impact on normal microflora in laboratory environments. There's been some promising research on bacteriocin usage, but it's been most extensive in food research. Finally, a self-plug for a recent publication. I just wrote a review on bacteriocins and their potential usage in conjunction with antibiotics, which is currently in press in IJAA. I love the podcast so much, Veronica. What's IJAA? International Journal of Antimicrobial Agents. There you go. All right, here's one from Andrea. Hi, TWiMitos. That's a new one. I like that. TWiMitos. Have you seen this? Unbelievable. So here's a woman who flossed her teeth and then developed a knee infection. And they isolated bacteria from the knee, and it was a common mouth bacterium. And it turns out that she had knee replacement surgery 5 years earlier. So that may be what's going on. What do you think of this, Michael?
I think it's quite possible. Remember, you become bacteremic each time you brush your teeth for about 4 hours. And so you're bacteremic 8 hours a day if you brush your teeth twice each day. And, knee
I thought that was a great argument for not brushing your teeth.
Only if you don't want to keep them. Only if you don't want to keep them. The issue is that the new joint serves as a nidus, and the bacteria can get trapped in the new joint, and they're effectively immunoprivileged, and your immune surveillance doesn't hoover them out, and the net consequence is you can get a localized infection, and that's indeed what this person had happen.
Is it rare, Michael?
It's rare.
Michael, do you believe in the prophylactic use of antibiotics before dental procedures?
No.
For people who have prosthesis?
No, I do not. The Brits have done exhaustive studies and showed, and I believe in evidence-based medicine, and the evidence from the United Kingdom shows that you don't need to necessarily prophylax before surgery in order to get good engraftment. What you do need to do is you need to clean the area, and that's why the evidence out there is in supporting chlorhexaphene bathing. Typically, if you're going to have joint replacement surgery, your orthopedic person will give you as a pre-op requirement that you bathe twice with these hexacorphine bathed soaps, and then you shower the morning of the surgery, again using hexacorphine, in order to reduce the load on your skin because they're effectively introduced.
I've had both my knees replaced, and I didn't do that. But on the other hand, I haven't had an infection. Knock on wood.
This evidence is relatively new within the last 3 years that they've really begun to prove that the hexacorphine bathing does work. My brother,
I had mine done. I had mine done quite a few years ago. So
my brother had his hip done last year at this time, and he actually came home with the bottle of soap and said, "Do I need to do this?" I says, "Yep, you got to take the bath." And he says, "I haven't had a bath in forever." I said, "Well, maybe it's about time." He says, "I shower." I said, "No, take the bath."
Andrea says it's 64 Fahrenheit and rainless in Seattle. Love the podcasts. Will writes, love the podcasts. Keep them coming. In response to Michelle bringing up the New York City Legionella outbreak, Vincent was trying to come up with Michigan-specific infections. Unfortunately, here in southeast Michigan, mostly in Detroit, we seem to have created a breeding ground for infections with highly resistant organisms, including carbanicillin-resistant enterococci, vancomycin-resistant Staph aureus, healthcare-associated Acinetobacter, and even a small outbreak of 5 cases of colistin-resistant, carbapenem-resistant Klebsiella. Some have suggested this is due to the high prevalence of diabetes, end-stage renal disease, as well as high baseline rates of MRSA and VRE infections in the area. All of these factors increase the probability that a patient will at some point require vancomycin or carbapenem to treat an infection. So, apparently multi-drug resistant organisms are Michigan's microbial gifts to the world. Anyway, as a medical student, I don't get a ton of time to enjoy our wonderful summer weather, 27°C today, so I'm going to do that while it lasts. Well, thank you, Will. And Michelle's not here, but next time we'll have to tell her that Michigan has its own problems.
I think since she teaches med students, she already knows and talks about that.
Don writes, pages 41 to 45 of Popular Science magazine, August 2015, discusses the danger of our war on bacteria and viruses. They cite hospital problems where 75,000 people die of hospital-acquired infections in spite of strong efforts to make things sterile. This war may be related to the rise of the superbug that is rendering our antibiotics useless. The space station is probably a dangerous place because of the lack of normal microbes. Probably other reasons, too.
Well, I got a letter from one of our listeners who sent me a paper on the impact of spaceflight on bacterial virulence and antibiotic susceptibility that I'm going to propose to the group here that we may discuss on an upcoming TWiM that will specifically address Don's question.
All right. Sounds good.
That's good. Sounds good.
One more is from Brian, who wrote to Elio. Elio's getting a lot of TWiM email. This article in the most recent issue of Scientific American, it's a special issue with a picture of Einstein on the cover, reminded me of the points you raised during a recent episode of TWiM. It has long been my opinion that the best advice to give to someone in school is to develop expertise in 2 complementary fields. In my case, I was able to combine skills in shipboard engineering and maintenance with at least a modicum of understanding of computer technology. Similarly, I think that someone with a degree in microbiology who is fluent in Portuguese, the Brazilian variant, or Chinese would be in a strong position. A person with a degree in microbiology who went to law school and specialized in intellectual property law could probably retire at 40. The attached article describes a need for a marriage between computer scientists and neuroscientists, and by extension with any of the biological sciences. That marriage sounds like a promising combination for a student, a major in microbiology and a minor in computer science, or vice versa. Yeah, this is absolutely true. Computer technology is absolutely needed to interpret the data we're generating, not just nucleic acid data sets, but any big data set. You need to have computer skills. And those students who get those skills are going to have an advantage. There's no doubt about that.
And statistics.
But I think that Brian wasn't aware of the fact that I think universities are quite aware of this. And it seems to me that a fair amount of energy and effort is being spent in trying to show what else people can do with themselves.
Well, I mean, I don't know about you, Michael, but in our micro program, we don't teach students
no
computer science, and we should. Now, maybe they're going to have it from undergrad increasingly, right? Because more students are getting exposed to it. But I think at least in the first year, they need to have a course that teaches them how to use programs to analyze sequences.
Advanced stuff. Yeah, everybody comes.
Well, that came up at the ASM focus meeting that we had on branches, and Chris Kellogg suggested that we develop workshops at our national meeting or our branch meeting specifically to teach programming language to microbiologists. And she suggested a couple of models, and so I'm going to actually suggest to our branches that we begin to offer sessions on various aspects about bioinformatics tools and actually learning how to program in R and in other things that Chris so eloquently made her impassioned case for. So I think Vincent's right on track.
Correct me if I'm wrong, but I think universities or departments of microbiology are giving opportunities for finding out about alternative careers. Law for people who want to become patent lawyers, business, engineering. It seems to me that this is happening. Am I wrong?
Oh, no, it is. It is.
Yeah,
and in fact, a number of our graduates have gone on to long and distinguished careers in patent law. Two graduate students who completed their degrees in the 90s have made partner at two law firms in the D.C. area.
It's happening, but not in a structured way, Elio, okay? And I think it has to happen. My department here doesn't do any of it. We don't tell people anything about alternative careers. I think that's a shame. I think NIH has to somehow build into its funding mechanism a requirement for that you have to have at least some way of educating students about alternative careers, because you can't just train them to be PIs, right? Because they can't all be PIs.
And that was in that paper that we discussed recently from Wisconsin.
That's right. Exactly right. Yeah.
And so I think
exactly what Bryant is saying, but it leads to it. Okay,
sure,
I think he's got a very good point. I think the synergy between backgrounds is so powerful that I know people who've done one thing their whole career very, very successfully, but they're kind of the exception. I think the people who do well are people who shift gears. When they go from graduate school to postdoc, they go into another field or something like that. That is very powerful stuff. I think Brian has a real good point.
So let me give you an example. I just interviewed a professor at Princeton who just illustrates this perfectly. Her name is Ileana Christea. She trained as a chemist and then learned mass spectrometry. Okay. She became fluent in mass spec and then became a virologist, and she is the best person in the world who uses mass spec to study virology. She does it in her own lab. So it's a perfect example of what Brian is talking about.
Yeah, blended skill sets.
So you don't have to ask for a collaborator to help you do mass spec. You just do it in your lab. All right, this episode of TWiM will be found at microbeworld.org/TWiM and also on iTunes. And if you have questions or comments, please send them to twim@twiv.tv. Elio Schaechter is at Small Things Considered. Thank you, Elio.
My pleasure.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
I'm Vincent Racaniello. You can find me at virology.ws. Our thanks to ASM for supporting TWiM and to Chris Kandian and Ray Ortega for technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.